The Retinol Binding Proteins as Antibodies by Genereux. As I discussed in Extinguishing the Fires of Hell, one common theme that cuts across all the autoimmune diseases and even breast cancer too is the comorbidity of eczema. At a more fundamental level, and what many others have reported on, is there's another cross-cutting syndrome, and it's that of insulin resistance. So much so that many people feel that insulin resistance is at the root cause of many of the chronic diseases. The very definition of type 2 diabetes is insulin resistance and metabolic disease. But it goes well beyond that. Insulin resistance is hugely implicated in coronary artery disease too, heart disease, and surprisingly even the brain diseases such as Alzheimer's. You've probably heard the Alzheimer's disease even being referred to as type 3 diabetes. So let's see if we can make some sense out of all this and find some plausible common ground and the connections between them. When I wrote about my theory of obesity causation back in October, I referenced this 2016 report documenting the adipogenesis effect of retinoic acid, circulating retinoic acid levels and the development of metabolic syndrome. I was somewhat expecting people to call me out on my use of that study. One of the weird aspects to it was even though the authors documented the adipogenesis effects of retinoic acid, they also concluded that the elevated levels of retinoic acid were beneficial in reducing metabolic syndrome. Quote, conclusions. The serum retinoic acid level is inversely associated with the development of METS independently of adiposity and insulin resistance. End quote. So, even though one aspect of that study supported my sub-theory of obesity causation, it directly contradicted my overarching theory that retinoic acid was the real culprit in causing the modern chronic diseases. Of course, there is an absolute mountain of other studies and research proving the incredible toxicity of retinoic acid. To me at least, something was not adding up here with their contrary findings. But it is rather easy to see where they go wrong in their analysis. Here's the source of the problem stated in the materials and methods section of their report. Quote, determination of retinoic acid concentrations. Serum retinoic acid concentrations were measured using a commercially available Elisa kit according to the manufacturer's instructions. This assay has high sensitivity and excellent precision for the detection of human retinoic acid. This assay has high sensitivity and excellent precision for the detection of human retinoic acid. End quote. Do you see it? They did not actually measure serum retinoic acid concentrations at all. Rather, they measured a biological proxy marker for it. Of course, there's no such thing as human retinoic acid either. So what are the Elisa kits? They are kits that test for and measure antibodies. The enzyme-linked immunosorbent assay, ELISA, is a commonly used analytical biochemistry assay, first described by Engvall and Perlman in 1972. The assay uses a solid-phase enzyme immunoassay, EIA, to detect the presence of a ligand, commonly a protein, in a liquid sample using antibodies directed against the protein to be measured. Performing an ELISA involves at least one antibody with specificity for a particular antigen. For example, you can use an ELISA test to detect and measure the antibody proteins to measles. ELISAs are not tests used to measure and quantify specific compounds such as retinoic acid. Using gas chromatography and mass spectrometry is the more standard technique for identifying and quantifying the molecular makeup of compounds. Of course, in the human body, most circulating retinoic acid, and retinol too, is bound up in wrapper retinol binding proteins. Therefore, what the authors meant to say was, quote, this assay has high sensitivity and excellent specificity for the detection of human retinol binding proteins for retinoic acid, end quote. So yes, if the body is building the needed retinol binding proteins fast enough, then it will safely wrap up the other highly toxic retinoic acid molecule. When wrapped up in the retinol binding protein, the retinoic acid will pose far less of a hazard and people are better protected from it. With this protective mechanism explained, we can now understand their observations and paradoxical conclusion. But the use of the ELISA test is not some major error on the part of the report authors either. It's probably a good surrogate test, and it appears to be a rather standard practice. Here's another report where they've done the same except it's for retinol binding proteins. Quote, retinol binding protein 4, and insulin resistance in lean, obese, and diabetic subjects. Measurements of serum retinol binding protein 4. Quote, serum retinol binding protein 4 was measured by an enzyme-linked immunosorbent assay, ELISA, ALPCO diagnostics, in group 1 and 3, and by quantitative Western blotting with purified human retinol binding protein 4 standard in group 2. Immunodetection was performed with a polyclonal antibody to human retinol binding protein 4, decocytamation, end quote. In addition to the ELISA, they are also quantifying their measurements with the Western blot test. But, like with ELISA, the Western blot is another type of test for measuring antibodies. Next, let's consider their results. Results, quote, serum retinol binding protein 4 levels correlated with the magnitude of insulin resistance in subjects with obesity, impaired glucose tolerance, or type 2 diabetes, and in non-obese, non-diabetic subjects with a strong family history of type 2 diabetes. Elevated serum retinol binding protein 4 was associated with components of the metabolic syndrome, including increased body mass index, waist-to-hip ratio, serum triglyceride levels, and systolic blood pressure, and decreased high-density lipoprotein cholesterol levels. Exercise training was associated with a reduction in serum retinol binding protein 4 levels only in subjects in whom insulin resistance improved. Adiposity, GLUT4 protein, and serum retinol binding protein 4 levels were inversely correlated. End quote. This is an important finding, as it is tying together serum retinol binding protein 4 levels, insulin resistance, diabetes, metabolic syndrome, and more. Except just for now, ask yourself, why are they using antibody tests to measure retinol binding protein levels? Doesn't something about that strike you as being a bit peculiar? To be clear, these are not procedural anomalies. It appears to be a preferred measurement technique. Here's another study where they use Western blotting to measure serum retinoic acid levels. Valproic acid combined with 13-cis-retinoic acid and 1,25-dihydroxyvitamin D3 in the treatment of patients with myelodysplastic syndromes. Of course, using mass spectrometry to measure retinoic acid levels is more problematic because the retinoid is entirely encapsulated within the retinol binding protein wrapper. Therefore, samples first need to be dissolved in solvent to break down the protein shell and wash out the embedded retinoids. From there, mass spectrometry can be employed. So although the ELISA and Western blot are measuring a biological proxy marker, they are probably more expedient tests and good enough for the purpose of their studies. Okay, now we need to really think about this. If these laboratory tests used to measure serum levels of retinoic acid, and even that of retinol, are in actuality antibody tests, isn't it pretty logical that the retinol binding proteins are indeed antibodies? Except for the last 50 years now, medical science has claimed that the retinol binding proteins are specialized transport proteins. The grand theory is that the liver first scrubs retinol out of serum and then sequesters it away into storage for later delivery. On an on-needed basis, the liver then releases the retinol wrapped up in the retinol binding proteins for transport. That's a nice sounding theory, but there's a huge flaw now showing up in that theory. That flaw is because a large percentage of the retinol binding proteins are actually being built and released by the adipose tissue and not just the liver. That's correct, the origin of much of the retinol binding proteins is from the adipocytes. Conclusions, quote, retinol binding protein 4 is an adipocyte-secreted molecule that is elevated in the serum before the development of frank diabetes and appears to identify insulin resistance and associated cardiovascular risk factors in subjects with varied clinical presentations. These findings provide a rationale for anti-diabetic therapies aimed at lowering serum retinol binding protein 4 levels, end quote. Okay, it looks like the grand theory of the retinol binding proteins being transport proteins is starting to fall apart. Quote, until 2005, the sole known function for retinol binding protein 4 was to mobilize retinol from tissue stores and deliver it to vitamin A responsive cells, where it can be converted to retinoic acid for use in regulating vitamin A dependent transcription and functions. In 2005, Khan and colleagues reported that circulating retinol binding protein 4 levels affect glucose clearance, with high retinol binding protein 4 levels inducing insulin resistance. Specifically, Kahn and colleagues propose that adipocyte-derived retinol-binding protein 4 is a signal that contributes to the pathogenesis of type 2 diabetes, linking obesity with type 2 diabetes, as well as other obesity-related metabolic diseases. End quote. Very importantly, this then brings into question the entire premise of retinol's storage in the liver. That's because the thought to be delivery proteins for it is being generated by and secreted from the adipose tissue and not at all exclusively from the liver's storage site. Next, let's think a bit about the cell response to viral infections. When a cell gets infected with a virus, it can very mistakenly start replicating and cloning more of the same virus. But very often, it also wraps the newly made viruses up in a protein capsid. Some cells can then reject the entire capsid out of itself. Secondly, in response to viral infections, the adaptive immune system will usually start to build antibodies to the virus. The goal and function of the antibody is to attach itself to specific binding sites in the viral protein. In doing so, it effectively bulks up any free circulating viruses so that they become too large to pass through the cell membrane. If successful, the viral infection is brought under control. Except now when a cell is contaminated by a toxin, it is by a non-protein based molecule. There are no protein binding sites at all to attach an antibody to. Therefore, as with some viruses, the cell needs to wrap toxic molecules in a specialized hollow core antibody or capsid if you prefer. Unlike the currently recognized Y-shaped antibodies, the retinol binding proteins are more barrel-shaped. That barrel shape is needed to engulf the entire antigen. Shown below is a model of the retinol-binding protein with its embedded retinol ligand. Structure of human holoplasma retinol-binding protein 4. Note the retinol molecule, ball and stick model, nestled inside of the far more complex wrapper, ribbon model, of the retinol-binding protein. Remember that retinol is too toxic to be in serum alone. Here's the spacefill model of the retinol-binding protein showing that no part of the retinol molecule is exposed outside of the retinol-binding protein itself. Therefore, this molecular structure is an astonishing feat of chemical separation and isolation. Not only has the cell's protein weaving machinery been able to detect and identify a single molecule, but it has also separated it out and wrapped it in a complex protein envelope. Remember too that nature is no fool and it is not wasteful. This extreme measure of molecular assembly and folding dexterity has to be going on for a very good reason. Why would the body resort to using such an elaborate and costly transport protein structure? Secondly, if this is truly a transport protein, then how does the receiving cell extract out the retinoid payload when no part of it is exposed to the outside world? And how exactly does it do so in a toxic-safe manner? Why does the retinol-binding protein only transport just one molecule at a time? And once again, why are the adipose tissues generating and secreting the retinol-binding proteins in the first place? Of course, no one knows the answers to these questions. At best, there are just some unsupported speculations tossed around about it. Quote, retinol-binding protein is also bound to a carrier protein, transthyretin. The process by which retinol-binding protein releases retinol for cellular availability is still unknown and not concisely determined. End quote. Additionally, if the retinol-binding proteins are transport proteins that are supposed to be so highly regulated, then why do they accumulate with age? Chart serum retinol level and BMI in USA. The real clincher question is, if the liver is building retinol-binding proteins to deliver retinol to the other tissues, then why are those receiving tissues then themselves wrapping up retinol in their own internally built retinol-binding proteins and ejecting it? The current theory of the retinol-binding proteins being transport and delivery proteins makes no logical sense. Isn't it just so much more logical that the retinol-binding proteins are hollow core antibodies? Isn't it just more logical that as the body gets exposed to more of a toxic load outside of liver storage, the body builds more antibodies to combat and protect us from the now adipose accumulating toxin? Here's another study indirectly indicating that's the case. Summary. Quote, cellular retinol, retinaldehyde, and retinoic acid binding proteins were localized in rat retina during pre- and postnatal development by indirect immunofluorescence. End quote. What is immunofluorescence? Quote, immunofluorescence is a technique used for light microscopy with a fluorescence microscope and is used primarily on microbiological samples. This technique uses the specificity of antibodies to their antigen to target fluorescent dyes to specific biomolecule targets within a cell, and therefore allows visualization of the distribution of the target molecule through the sample." End quote. Clearly, the retinol-binding proteins are very antibody-like. But, since we now know they are not transport proteins, is it not highly likely they are indeed antibodies? They have just not yet been recognized as such. The next logical question is, if the retinol-binding proteins are, in actuality, antibodies, then what does that tell us about retinol? It tells us that it is a toxin. The famous amyloid plaque of Alzheimer's disease. One of the key neuropathological hallmarks of Alzheimer's disease is the accumulation of beta amyloid plaques in the brain. No one knows why they develop or what to do about them. What's assumed is that they are just defective ribbon-structured proteins that exhibit some weird and unexplained misfolding. But doesn't that sound a lot like the folded ribbon proteins of the retinol-binding proteins? Here's a description of the retinol-binding proteins. Lipocalins. Quote, "Members of the lipocalin family, including the retinol-binding protein, RBP, epididymal retinoic acid binding protein, and beta-lactoglobulin, share a very low sequence identity but display a highly conserved overall fold. They're composed of an 8-stranded antiparallel beta sheet that is folded over itself to form a hydrogen-bonded beta barrel, which constitutes the ligand-building pocket, figure 2. The N-termini of these proteins are folded around the back of the barrel, capping that side of the pocket." End quote. Next, we need to know and deeply appreciate that it's not just the liver and adipose tissues that are excreting the retinol-binding proteins. It's also happening in the kidneys, lungs, heart, skeletal muscle, spleen, eyes, and testes too. Quote, "Functions of retinol-binding protein. Retinol is secreted from its storage pools and circulates in blood bound to retinol-binding protein. The main storage site for vitamin A, and correspondingly, the main site of the synthesis of retinol-binding protein, is the liver, although other tissues, including adipose tissue, kidney, lung, heart, skeletal muscle, spleen, eye, and testes, express this protein." End quote. And then there's this statement, quote, "The mechanism by which retinol initiates secretion of retinol-binding protein from cells is unknown, but appears to be conserved in yeast ectopically expressing retinol-binding protein." End quote. I think it is quite obvious that the cells of multiple tissue types are synthesizing their own retinol-binding proteins as a protective measure in an attempt to rid themselves of a highly toxic molecule. Do you see where I'm going with this? Yes, if the cells of the liver, adipose tissues, kidney, lung, heart, skeletal muscle, spleen, eye, and testes are all able to secrete retinol-binding proteins with the retinoid wrapped up inside of it, why shouldn't we think that the nerve cells of the brain are not going to do the same? Just like the fact that the retinol-binding proteins are unrecognized as antibodies, couldn't the famous amyloid plaques of Alzheimer's disease be unrecognized as retinol-binding proteins generated by the brain too? Or maybe, since Alzheimer's is such a new phenomenon in human existence, the amyloid plaques are an ongoing attempted synthesis of retinol-binding proteins. Thinking that the cells of the nervous system can generate amyloids is not just speculation. As mentioned above, the retinol-binding proteins are often bound in serum to transthyretin. Quote, "Transthyretin, formerly known as prealbumin, is a plasma protein involved in the transport of thyroxine and retinol and is secreted in great amounts by the choroid plexus epithelium." End quote. Knowing that transthyretin is typically bound up with the retinol-binding proteins, it's no surprise that so many people with diabetes, autoimmune diseases, and even dementia have the comorbidity of abnormal thyroid functions. There are some rather remarkable similarities between the so-called defectively folded proteins of the amyloid plaques and that of the folded over proteins of retinol-binding proteins. For example, here's the crystal structure of the protease inhibitor domain of Alzheimer's amyloid beta protein precursor. As with the retinol-binding proteins, the Alzheimer's disease defining proteins adopt a beta sheet structure and sometimes form into amyloid plaques.
Most importantly, when you consider that there has been an 800 times increase in the rate of Alzheimer's disease since the early 70s, it is completely, absolutely, undeniably, positively crystal clear that the disease is a poisoning. It's then easy to appreciate that the cells of the brain are responding to that poison in very similar ways as do, say, the cells of the kidneys, lungs, etc. However, once trapped inside the brain, there is no viable disposal pathway for these unusual proteins. The brain just slowly accumulates and clogs up with the amyloid plaques. Not only that, but all the energy and resources wasted in building the amyloid plaques has been taken away from the brain's normal tissue maintenance and repair processes. Very tragically, billions of taxpayer-funded dollars have now been spent by researchers going on a wild goose chase of blaming genetics for this disease. Of course, it is a colossal waste of money and time because it is completely obvious Alzheimer's disease is not caused by genetics. Not only does this take thousands of years for a genetic shift to occur in the human population, the country with the highest genetic diversity on the planet now has the highest rate of Alzheimer's disease in the world. And who cares about some supposed genetic predisposition to the disease because there's absolutely nothing people are going to be able to do regarding their genetic makeup. So, investigating genetics is pretty much a meaningless distraction from the real culprit of the disease being environmentally caused. If people were not being slowly poisoned by some environmental factor, then their genetic predisposition would never be a problem. Of course, it's not just the brain that's clogging up with amyloid plaques. It's the heart and other organs too. Not at all surprisingly, that also directly correlates to the serum levels of the retinoid-binding proteins. Identification of transthyretin cardiac amyloidosis using serum retinol-binding protein 4 and a clinical predication model. Quote, findings in this case control study that included 34 patients with transthyretin cardiac amyloidosis and 77 control participants. Retinol-binding protein 4 was significantly associated with the disease independently of tested confounders. A prediction model composed of retinol-binding protein 4, transthyretin, and echocardiographic and electrocardiographic characteristics had excellent discrimination for transthyretin cardiac amyloidosis, deposition of amyloid, end quote. Conversely, the vascularization of the brain in Alzheimer's dementia patients often shows the same type of damage as that presented in coronary artery and heart disease. The condition is called vascular dementia. Therefore, it's very probable that the same root cause, force, behind that vascular damage is driving both diseases. Equally important is to know that almost no one dies directly from Alzheimer's, rather they die with Alzheimer's. Meaning the real cause of death is one of their other conditions, such as kidney disease, heart disease, stroke, choking, pneumonia, etc. Additionally, it's well documented that persons with type 2 diabetes are a bigger risk, 200%, for developing Alzheimer's disease. It is even claimed that the single biggest risk factor for developing Alzheimer's disease is having type 2 diabetes. Therefore, many people have made the erroneous assumption that the diabetes is somehow causing Alzheimer's disease. But that's not quite true. Diabetes is not causing the Alzheimer's. Rather, both diseases are simply joined at the hip, metaphorically speaking. And in both diseases, we have the common themes of insulin resistance and the development of amyloid plaques. Obviously, diabetes is not causing the Alzheimer's, and Alzheimer's is not causing the diabetes. Once again, something else is at the root cause of both diseases. As we've seen, elevated serum levels of retinol-binding proteins are a significant marker in both diseases too. It is also true for the development of amyloid plaques of the heart. So much so that it is pretty much an undeniable fact that the retinol-binding proteins are somehow involved. But there is an even more direct connection between these two major diseases. As with heart disease, it's the accumulation of misfolded proteins. Protein misfolding and aggregation in Alzheimer's disease and type 2 diabetes mellitus. Quote, Alzheimer's disease is characterized by the accumulation of amyloid beta in the brain, while type 2 diabetes mellitus is characterized by the deposition of islet amyloid polypeptide, IAPP, also known as amylin, within beta cells of the pancreas. End quote. And then a bit later they state, quote,
It is becoming increasingly believed that isolate amyloidosis is the progeny of many diseases, including type 2 diabetes mellitus. The production of isolate amyloid polypeptide, IAPP, oligomers that result in amyloid deposition is considered as a major contributor in pathogenesis and progression of type 2 diabetes mellitus. This has been found present in 96% of type 2 diabetes mellitus patients and is recognized as a hallmark for diagnosis of this disease, end quote. And then, linkage between Alzheimer's disease and type 2 diabetes mellitus. Quote, Alzheimer's disease and type 2 diabetes mellitus are both prevalent in the aged population. Whereas the cerebral accumulation of amyloid beta is a major pathological hallmark of Alzheimer's disease, the deposition of a very different polypeptide, amylin, that likewise succumbs to beta sheet formation and self-aggregation, occurs within the pancreas, especially in beta cells in type 2's diabetes mellitus, end quote. So now, just what do you suppose is responsible for causing the messed up structure of the proteins generated by the pancreatic and other tissue stem cells? How about we seriously consider it to be a toxic molecule that has now been proven to cause about 500 different gene expressions in stem cells? Except to most of us here, it's now clear these are in actuality sites of gene damage and not that of gene expression at all. That gene damage then messes up the cell's protein weaving machinery. Naturally, the follow-on consequence is going to be the defective structures of the manufactured proteins. Somewhat like with the wild goose chase of genetics, there's been a vast amount of research in trying to correct the misfolding proteins using pharmaceutical drugs. Of course, that effort has been futile. Additionally, most research is now narrowly focused right down into trying to decipher what's wrong with the protein structure. Oddly, it appears most people are only asking why the protein is misfolding, as if something has gone wrong with it post-manufacturing. They are not considering that it has been expressly manufactured that way due to RNA-DNA damage. The retinol binding proteins tied together with insulin resistance. GLUT4 is the glucose transport protein used by cells to take up insulin from serum. Obviously then, if the GLUT4 protein are not of sufficient quantity or quality, it then will cause the condition of insulin resistance. It's not hard to imagine that if the cell's energy and resources are tied up generating retinol-binding proteins, that could be impairing their production and the functioning of the GLUT4 proteins. This scenario is indeed being demonstrated in clinical research. For example, serum retinol-binding protein 4 contributes to insulin resistance in obesity and type 2 diabetes. Quote, we show that serum retinol-binding protein 4 levels are elevated in insulin-resistant mice and humans with obesity and type 2 diabetes, end quote. This now brings us full circle to where I hope it shows how it's all interconnected. The information presented here not only links the retinol-binding proteins to type 2 diabetes and the amyloid beta plaques, it seriously implicates it in causing coronary artery, heart disease, and of course Alzheimer's too. Except, we need to be very careful here and not try to blame the retinol-binding proteins. Rather, it's the overload of retinol that is the very root cause. It's the overload of retinol that has forced production of the retinol-binding proteins in the first place. There's one more connection with insulin resistance that I'd like to discuss, and that is of mitochondrial dysfunction. There are hundreds of research papers documenting that mitochondrial dysfunction and diabetes are very closely coupled. But, as always, there's more to the story, and that is the key role of the retinoids directly damaging the mitochondria. Vitamin A and retinoids as mitochondrial toxicants. Quote, overall, such findings indicate a potential ability of vitamin A and its derivatives to negatively interact with biological membranes, an event that may lead to organelle stress, as for instance, mitochondrial dysfunction, and to cell apoptosis or necrosis, end quote. And, quote, vitamin A and its derivatives, the retinoids, disrupt mitochondrial function by a mechanism that is not completely understood. However, it accounts with impaired electron flux between the complexes of METC, increased reactive oxygen species production, and induction of oxidative and nitrosative stress to mitochondrial membranes. Additionally, vitamin A and retinoids alter the mitochondrial structure by causing swelling of the organelle.
More investigations are needed to elucidate how vitamin A and retinoids affect mitochondria and whether there is a causative link between such events and the clinical manifestation observed both experimentally and in humans." End quote. Chart: in vivo effects on mitochondrial membranes. Predictably, the thinking from some pharma-funded researchers is the same. It's viewed as an opportunity to try to develop a new drug that will block or intervene in the synthesis of retinol-binding proteins. Of course, it always comes down to needing a drug to block something. Using a non-drug-based therapy is completely unthinkable to these folks. But since we know with 100% certainty that Alzheimer's is the result of a long-term poisoning, the only drug that could even possibly work is one that acts as an antidote to whatever that poisoning is. Until then, almost all other drugs are just going to be useless, and most will cause even more harm. Now, with the known connection between the elevated retinol-binding proteins and the development of amyloid plaques, and having a better understanding of retinol-binding proteins likely being an antibody response to a toxin, isn't it very likely retinol is the responsible toxin? Maybe it's just me, but how about we just consume a lot less of these retinoids and let the body heal itself? Chart: Alzheimer's, Atlantic provinces, and Crohn's rates, Nova Scotia. Are the retinol-binding proteins antibodies? Please have a think about it and comment as you see fit.